It's my great pleasure to introduce her too. So Allison is also at the Institut Curie in the Department of Developmental Biology and Genetics. And she has been working for many years now on the fruit fly system, trying to understand what happens in terms of stem cells in the gut, and using all of the elegance that Drosophila genetics and cell biology can give. And she has many different avenues of research. Today she's going to be telling us about Chromatin State Transition Modelling. She has studies on aging as well, and all sorts of other things. But I look forward to hearing you, Allison. Thank you so much.
Okay, great. And I'd like to start by reiterating what Sylvia said. I'm really excited to be here live after over a year of talking on Zoom and in my living room. So that's really fun. So today, yes, as Edith said, I'm going to tell you about stem cell lineage decisions, and in particular, the role that chromatin remodeling and chromatin state transitions play on that. And before I discuss stem cells, I would like to first take a step back and begin at the larger scale of adult tissues. And as tissue growth and tissue renewal is something that's critical during both embryogenesis as well as our early life and adult life, you can also imagine that as we age becomes really critical, as was mentioned both in Leila's talk and other talks. And we can also just imagine that this process must be also important in other animal species, such as those that are quite long-lived, including the Asian elephant, which can live upwards of 90 years of age, or the bowhead whale, which can live over 200 years of age. However, it's not only in these sort of exotic species that we can study this, thankfully, but we can also look at this in both the mouse as well as the fruit fly, which is the model system that my lab uses. So, of course, adult stem cells are critical for this process of tissue renewal. They divide, they self-renew. They will go on to produce daughter cells that can terminally differentiate and replenish those cells that have been lost in the tissue. And this process is called differentiation, of course. And I think what's really fascinating is that you go from a cell that has these self-renewing capabilities to a very different type of cell that has very specialized metabolic functions. So this really raises the question of what changes arise and how are these changes regulated. And we know that, for example, gene expression changes are critical for this. So, for example, we have gene A, that's a stem cell specific gene. It needs to be active in the stem cell. It needs to be turned off as the cells differentiate, and genes B and C need to be turned on later during the differentiation process. So how is this regulated? And of course, RNA polymerases play a major role here. They need to bind to the promoter and activate genes. Transcription factors also bind to enhancers and promoters and will drive gene expression. But chromatin modifying enzymes as well as remodeling factors are crucial to control the accessibility of transcription factors to the DNA. And work from Edith's lab and those of other labs in recent years has also demonstrated that the 3D architecture of chromosomes, as well as the nucleus in and of itself, are also critical to control gene expression changes. We also know that this process is critical not only during a normal lineage differentiation, but it also has a cancer preventative function, so that it's been demonstrated that modification of epigenetic marks, for example, can allow cells to reacquire pluripotency or to acquire characteristics that are essential to drive tumorigenesis. Okay, so the question that I would like to discuss today is really how do chromatin changes guide adult stem cells, both the self-renewal process as well as differentiation. So this is the model system that my lab uses. It's the adult intestine of the fly. So there are around 1,000 multipotent stem cells. They're shown here in red. They divide and produce two types of terminally differentiated cells, enteroendocrine cells that are in green, and enterocytes in blue, so much like the mammalian intestine. They have similarities not only to mammalian intestinal stem cells, but also other types of mammalian stem cells, such as those in the airways or the lungs and the skin as well. So we use this really as a model system just to understand stem cell biology in general. And of course, some of the advantages of this model are really the rapid lifetime of the fly, so going from an egg to an adult in 2 weeks of age. the rapid aging process, which, as Edith mentioned, my lab is also studying this process, and it takes place over a period of about 8 weeks of life. So that's quite convenient, as opposed to the geriatric mice that Chaire was telling us take 28 months to age. And of course, we have really extensive genetic tools to precisely inactivate or overactivate genes in cell type and tissue specific manners. So this is quite a simple stem cell lineage, actually. The stem cell divides to self-renew. It can then produce 2 types of precursor cells, one that is an enteroendocrine precursor that will divide once and make secretory cells, and the other, which is called an enteroblast, that will directly differentiate into enterocyte cells. So what was known for many years, actually, is that environmental signals in the intestine can drive the stem cell to proliferate. And these stem cells are really exquisitely in tune to changes in the environment, so cell death, for example, or any type of damage to the tissue. And there are many signaling pathways that become activated and coordinate that. I'm just going to highlight 2 of those that are quite important, which is EGFR signaling and JAK-STAT signaling. So a number of years ago, we set out to try to identify additional mechanisms that could also regulate stem cell self-renewal. And to do this, we wanted to do an unbiased genetic screen, which is something we can do in the fly. So we wanted to, for example, identify genes which, when inactivated, would lead to excess numbers of stem cells. Okay, so here would be our wild-type lineage, and here we would make overproduction of stem cells. This is what a wild-type lineage looks like, where we can mark a single adult stem cell, allow it to divide, and all of its progeny are marked by GFP expression here. The stem cell is marked by this red staining here. And one of our mutants is called Kismet, or it's the fly ortholog of CHD7 and 8, which I'll tell you more about in the next slide. But when this gene is inactivated, we now have this large expansion of these number of red cells, which are these stem cells. So what is Kismet and what are CHD7 and 8? So Kismet was originally identified as a dominant suppressor of polycomb, and inactivation of Kismet itself leads to homeotic changes in the fly embryo. It encodes a really large protein that is around 5,000 amino acids, and it has similarities to that of its mammalian counterpart, CHD7 and 8. They have these ATPase domains that's thought to slide nucleosomes, and other domains that really have quite ill-defined function. There were some studies that had tried to investigate sort of where is CHD7 binding in the genome and what might it be doing. And from those, it was shown that it's really localized to both the enhancers and promoters of genes that are active. So it was thought to promote gene expression. And interestingly, human patients that have one copy of CHD7 inactivated have a developmental syndrome called Charge syndrome, where they have defects in neural crest-derived tissue. So they have nervous system defects, hearing defects, visual defects. CHD7 is also a gene that is found mutated frequently in lung cancer. So it seems to have important roles in humans. And it was also demonstrated that in mouse, it's required in neural stem cells for proper neurogenesis. But outside of that, the adult stem cell specific functions and its downstream targets were not really well understood. So Louis Gervais, who's a senior scientist in the lab at the CNRS, he set out to try to understand what is Kismet doing, what is it doing in the genome, and how in particular is it impacting adult stem cells. So here, what he wanted to ask is the differentiation process deregulated as well? And he found that it wasn't, in fact. So while we see this massive accumulation of stem cells, if we quantify the ratio of the 2 different types of differentiated cells that are produced, they're really not disrupted here. So this suggests that Kismet is causing a defect in self-renewal, but that differentiation is actually happening normally. And that can also be seen in this sort of long-term experiment where we make either wild-type lineages or those that are mutant for Kismet. And you can see that these mutant Kismet lineages outcompete the wild-type tissue in the gut. So here, the whole gut is pretty much becoming green, as opposed to wild-type, where we just have these patches of cells. So this, again, is reflecting the fact that we have this massive overproliferation, a self-renewal process of the stem cells. So the question that we really wanted to understand is, why are these mutant stem cells overproliferating? And to address that, we wanted to identify what its genomic targets were and try to ask in a more global way, what is it doing in the genome? And so for several reasons, we turned to a technology that's called targeted DAM-ID. And this is somewhat unusual, so I'm just going to walk you through how this works. And this technology was developed by Andrea Brand's group, and this was based on previous studies by Bas van Stinsel and Steve Hennekoff, who had developed this technique in cell culture. And Andrea Brand really moved this into using it actually in vivo in the fly. And the way that this works is that you make a transgene that encodes your fusion, a gene encoding your fusion protein of interest, a chromatin factor, a transcription factor. This is then directly fused to a DAM methyltransferase that comes from E. coli. And this DAM can methylate the genome at GATC sites, which can then be digested and processed and mapped onto the genome to have a proxy for where your factor of interest is binding. So importantly for us, this allows us to map Kismet without an antibody that works for, say, for ChIP-seq. It can be done in a cell type specific manner, so we can do this specifically in the stem cell. It does not require cell sorting, and we can do this with low numbers of cells. So Louis did the following experiment. He compared DAM alone, which is our control, with a DAM that's fused to Kismet. And this is expressed at extremely low levels in the stem cell. And now I'm just showing you what the binding of this looks like across the genome. I'm just going to show you one of the genes that's important in the stem cell, which is the ligand delta, ligand of the Notch pathway. That's really a stem cell specific gene. And you can see that Kismet is found bound, accumulating at the promoter and in the gene. So where is Kismet located at a genome-wide level? So what Louis found is that if we look at genes that are active in the stem cell, Kismet is enriched at these genes as compared to genes that are silent or not expressed in the stem cell. And this is somewhat similar to RNA Pol2, which is also enriched at active genes, obviously, but different from polycomb, which is a repressor factor. If we then look at enhancer elements, and these are enhancers that have been defined experimentally by work from Alexander Stark's group, and he's classified enhancers as either developmental enhancers or housekeeping enhancers. And we found that Kismet is really specifically enriched at developmental enhancers as opposed to housekeeping enhancers, which is different from RNA Pol2, which is somewhat more enriched at housekeeping enhancers. And because of this link with enhancers, we wanted to test whether another enzyme that's critical for enhancer biology might also be playing a function in stem cell, in limiting stem cell self-renewal. So this is another complex, which is called trithorax-related complex in flies. It's in mouse or humans, it's the ML3-4 complex. And it has many activities, but one of its primary activities is to monomethylate histone H3K4, and it's required to activate enhancers, although that seems to be independent of its methyltransferase activity. So what we did was to test the inactivation of components of this complex, which is shown here. And what Louis found is that, indeed, if we inactivate this complex, we see a similar expansion of the number of stem cells, which you can see by this red mark here. So we have a very similar phenotype to that we saw through inactivation of Kismet. So this led us to wonder whether there may be coordinated action of Kismet and trithorax related, at least in terms of some of the downstream targets that they regulate. And this was interesting given some of what we know about the human pathologies associated with inactivation of components of ML3-4. So there are several components that are, when inactivated, are classified as a human syndrome called Kabuki syndrome. And interestingly, this syndrome really has a lot of clinical overlap with that of CHARGE syndrome, and they're often misdiagnosed in the clinic. So we wondered whether in both, perhaps in flies as well as in humans, they may have similar overlap and be important to regulate some of the critical downstream target genes. And one way that we wanted to test that was to ask where are they binding in the genome? Do they have overlap at the level of their targets? So we mapped where trithorax related was binding and compared this with that of Kismet, and we found that indeed they have a large overlap in terms of their genomic binding. We then, of course, wanted to ask which genes become deregulated when we inactivate Kismet or trithorax related, specifically in adult stem cells. So for that, we downregulate them using RNAi in the stem cell. We then FACS sort out the stem cells and purify the RNA and perform RNA-seq. And similarly, we found that they have quite an overlap of the genes that become deregulated upon inactivation of trithorax related or Kismet. And this is a larger percentage of the Kismet genes, as trithorax related has more sort of genomic influence than Kismet does. So then the critical question became really, is there a gene that is both bound by both of these factors and deregulated that can explain this increase of stem cell self-renewal? And we found a couple of genes, and one that caught our interest was a gene called CBL, which encodes a negative regulator of EGFR. So it was bound by both Kismet and trithorax related, and it was downregulated in both of these RNAi contexts in the stem cell. So could CBL be the downstream target of interest? And one prediction of that would be if we inactivate CBL, this should lead to an over-proliferation of the stem cells. And that is indeed what we found. So here in green, we are inactivating CBL. And we found that, consistent with some previously published work of Bruce Edgar's group, that this can lead to an expansion of the number of these stem cell-like cells here, which is very similar to the phenotype of both trithorax-related and Kismet. So CBL is an E3 ubiquitin ligase that promotes the downregulation of the EGFR receptor itself. So again, the prediction of this would be that upon inactivation of CBL, there should be an increase of the protein levels of EGFR receptor. So is that true, and is that also what we see then in Kismet and trithorax-related knockdown? So this is just what a wild-type stem cell looks like. Here we have EGFR, which is shown in red. Upon the downregulation of CBL, we see that we have an accumulation of EGFR at the membrane of the stem cells. And this is short-term downregulation of 3 days. And here, if we knock down Kismet, we have quite a similar phenotype where we have an enhanced expression or enhanced localization of EGFR receptor at the membrane. And we also find this in trithorax-related, although it's somewhat lesser than that seen in Kismet. So we think that altogether these data are consistent with the idea that these genes regulate CBL, which in turn regulates EGFR receptor. When there's more EGFR receptor present at the membrane, it can bind to the ligands that are present around the cell. So here's just the model for how we see that working in a normal context. We see that trithorax-related and Kismet both bind to CBL. They're required to produce a lot of CBL to allow for stem cell self-renewal to be limited through the downregulation of the EGFR receptor. In the mutant contexts, we now make less CBL, which now leads to an increase of EGFR protein and an enhanced stem cell self-renewal. Okay, so what I've told you so far is that we have identified Kismet and trithorax related as negative regulators of stem cell self-renewal through EGFR. What I'm not going to have time to go into is that we also have another factor of SPEN, which similarly blocks stem cell self-renewal. It also is an important transcriptional regulator. And what we wanted to address next was really how is chromatin organized in adult stem cells in a more global manner, and how does this organization change during the differentiation process? And actually, there are several studies to date that have approached this question. A majority of them have been in an in vitro context of stem cells that are differentiated in vitro, or in a developmental context. And here we wanted to actually look at this in an adult lineage that we felt would be under the influence of the external environment. We also wanted to take advantage of the fact that this was this had two different lineages, really, the enteroendocrine lineage and the enterocyte lineage, to ask, are the changes that happen in both of these the same, or are they different? So in particular, and I should say this work was done by a very talented PhD student, Manon Josserand in the lab, who's working with Louise Gervais on this project. And what Manon wanted to do was to ask, what are the chromatin state transitions that are associated with differentiation? So what do I mean by that? So again, if we have a gene A, which is a stem cell specific gene that needs to be turned off within both of these lineages, by what means is this gene being turned off? And in turn, how is gene B, which is a lineage-specific gene, how is this gene kept off in the stem cell and turned on in this lineage and kept off in this lineage? And finally, we would like to know what the relevance of the chromatin state changes are on the lineage specification and on the final cell state. So in order to address this question, we wanted to borrow from work of others. that have really used a reductionist approach to classify chromatin. And I should say that the classic model of chromatin is as a repressed state or an active state. And over the years, through mapping the binding of different histone marks and chromatin factors, it got a lot more complex. So there were factors that bound in various regions of the genome. However, a number of years ago now, around 10 years ago, there were several groups that had the idea to really reduce this number again. And I'm just going to focus on some of the studies that were done in Bas van Stinsel's group by Guillaume Fillon. And what he did was to map at a genome-wide level 53 histone marks and chromatin factors, and then to use a mathematical modeling approach to reduce these into a limited number of states. And what he found was that in their cell culture model, they could identify 5 major chromatin states, so 2 of which were active states and 3 of which were repressed states. So importantly, each of these states is really a combination of many histone marks and binding proteins, for example. And these states can be kind of recapitulated through representative factors. So for example, in these activated states, Brahma is important for this red state, and this seems to be a state that's more enriched at developmental genes, and it has higher levels, for example, of H3K4 trimethylation. Housekeeping genes are thought to be more in a yellow state that's bound highly by RNA Pol2. He could identify 3 major classes of repressed chromatin. One that they're calling a green state that is enriched in heterochromatin protein 1, HP1, as well as H3K9 di and trimethyl. Another repressed state that is enriched in polycomb, which is associated with H3K27 trimethyl. And interestingly, they characterized a sort of previously unknown silent state that they call black chromatin that is devoid of many marks, but is somewhat enriched in the linker histone H1. And work from Andrea Brandt's group has actually made transgenic lines that could express representative factors of these different colors of chromatin, as they were called. And what we wanted to do was to take advantage of these and to map where these are binding in the homeostatic adult intestine. So Manon did the following experiment, which was to use cell-specific drivers, and we could specifically express these various reporter lines in the stem cell, the enteroblast, the enterocyte, or the enteroendocrine cell. And unfortunately, we're lacking this cell type here, which does not currently have a good driver line. So we map all of these factors in these different cells within the lineage. And Manon did triplicates of all of these and performed the genomic sequencing of the methylated DNA that was bound by these factors. I'm just going to show you what the sort of raw data looks like for that. So here, if we look at a portion of the genome where we're going from a heterochromatic region into a euchromatic region, you can see that HP1 is really enriched in this heterochromatic region. And when we get into the euchromatin, now we have binding of Pol2 and Brahma as well as polycomb. So Manon, however, didn't want to just use the brute output of these data, but she wanted to, again, go back to a sort of a modeling approach. And for that, she teamed up with Natalia Rubinova, who has mathematical training and is a bioinformatician in the lab. And in collaboration with Owen Marshall and the University of Tasmania, they performed hidden Markov modeling on the binding of these factors in all of the cell types that we could profile. And I'm just showing you the 35-state model that we did in the intestinal stem cell, but we have the same data for the other cell types. So from this, they could find that there were approximately 7 major chromatin states. And again, these have many similarities with those which were previously described, which was actually what we were expecting. So we could identify 2 active states, again, a yellow, which is sort of enriched in RNA Pol2. It's lower in Brahma. A red state that is trithorax associated, that is enriched in Brahma as well as Pol2. This tends to have more tissue-specific functions or developmental functions than the yellow, which is more housekeeping roles in general. We could also identify 2 states that were intermediate, one that we're calling a polycomb mixed state because it has both polycomb as well as RNA Pol2. And we think that this state is actually, it's probably many of these genes that are in this state are actually likely due to heterogeneity within our population, where some cells may have these genes in an active Pol2 bound state, and others may have them in a polycomb repressed state. But we can't exclude that some of these genes actually may be true binding of both polycomb and Pol2. There's also another repressed state that's we're calling yellow repressed that has low levels of Pol2, and it seems to be primarily housekeeping genes that are broadly expressed. And finally, we can identify the three primary classes of repressed chromatin: a polycomb repressive state, that's blue, this HP1 associated green state, and this black state. Okay, so now if we plot these different states along the genome, I'm just showing you what these data look like for a portion of the genome in the stem cell enteroblast and the two differentiated cell types. You can see that overall it looks quite similar, which is not too surprising. But if you start to look more carefully, you can see that there are plenty of regions where we actually see quite a bit of differences that happen from the stem cell to the differentiated cells. So Mano really wanted to be able to look at these data in general and trying to understand how they were changing. And in order to simplify that process, she wanted to assign one gene a given state. So the way that this was done was to take the primary state that covers the length of a gene. So here is just an example of how we did this. So here is the gene Prospero, which is an enteroendocrine specific gene. It's primarily coded by this red state, so it has been assigned this red or trithorax group state. In the other cell types, this gene is we know from many other studies that this gene is not expressed, it's not active, and indeed we find it in a polycomb repressed state in the stem cell and enteroblast, and in a black state in the enterocyte. So if we just look at some cherry-picked genes out of the lineage, we see that basically they behave the way we would expect, so I'll just walk you through some of these. So delta, as I mentioned before, is a stem cell specific gene. We see that in the stem cell it's in this yellow state and it's repressed in these other cell types. Escargot is a gene that's specific for the stem cell and enteroblast, and it's in this red active state in the stem cell and enteroblast, and becomes turned off by a polycomb mixed and polycomb repressed state in these other cell types. Similarly, PDM1 is an enterocyte-specific transcription factor. It's found in a yellow state in the enterocyte and in a repressed state in the other cell types. And again, Prospero is in this red state in the enteroendocrine cell and off in the other cell types. So what Mano was really interested in was trying to take these data and describe in a general way what are the primary transitions that are happening, what are the primary ways in which a gene is going from an active state to a repressed state, and what are the genes that follow different trajectories? What are genes that go from, for example, from a yellow state to a green repressed state? So she first, we have lots of data, I'm just going to show you a little bit of her data. And she first wanted to focus on genes that go from an active state in the stem cell to a repressed state in differentiated cells. And what you can see here is that in the going from the stem cell to the enteroendocrine cell, the primary state that happens is going from active states to a green repressed state. And this is the HP1 associated state. This is somewhat different if we look at the enterocyte lineage. The primary change is going from an active state into a black state. But I also told you that in this lineage, we go from a stem cell to an enteroblast to an enterocyte. So what does this look like if we now put the enteroblast in the middle here? And that was interesting because what we found is that a majority of the genes that go from this active state to a black state actually pass first through this green state. And we don't exactly know what that means. One possibility is that this change needs to be very rapid, and perhaps there needs to be a sort of a K9 or HP1 associated chromatin that would be established before these genes would be shut off into this black state later in the lineage. So Mano also wanted to ask what are genes that go from a repressed state in the stem cell to an active state in the differentiated cells. And I'll come back to it in a second, but as you can easily see, that the primary transition we see is from this black state into an active state. But I'll just spend a second to talk about this polycomb repressed state, which there are very few genes that undergo this change from a polycomb repressed state to an active state. But interestingly to us, the important lineage regulators were found in this category. So prospero, as I mentioned, is really the critical transcription factor for the enteroendocrine lineage, and it is found here along with two other transcription factors that have been shown to be important for lineage specification here. And PDM1 is the key transcription factor for the enterocyte, and it's found in this polycomb repressed state in the stem cell before it becomes activated. And this is consistent with what others have shown in the field about polycomb really being important to regulate transcription factors. As I said, one of the primary states that caught our eye was really this transition from this large category of black states in the stem cell to activated states in the differentiated cells. And if we look at these genes and we perform gene ontology on these genes, we see that those that go from black to active in the enteroendocrine lineage have these types of terms, such as neuropeptide signaling, peptide receptor activity, etc. And these are really what we know about the hormone signaling capacity of the enteroendocrine cells. So this is really the metabolic program of the enteroendocrine cells. And similarly, in the enterocyte lineage, those that are going from black to active have terms associated such as proteolysis and fatty acid metabolism, which is really the enterocyte-specific metabolic program. So we think that this transition from black to active is really important for these physiology-related genes, and it's something that we're quite interested in. So that's where we are right now for the moment. I'm just going to give you a flavor for what we're using these data for and what is currently ongoing. So we're using these primarily in two different ways. The first is that I told you that we know that genes that are really critical for the lineage, for example, are following this polycomb repressed state to an activated state. So we're asking what other genes are also found that undergo this transition, and could they have important functions either for lineage specification or could they be important stem cell regulatory genes? And she has several genes that are quite interesting that she is further characterizing through genetic manipulation that seem to be transcription factors, for example, stem cell specific transcription factors. So we're asking, do these genes play critical roles in the stem cell or on lineage differentiation? And the other question that we're quite interested in is really trying to understand how these transitions occur and if the transition themselves are actually important for the final differentiation process. And one that we're focusing on is this black to active state. And the approach that we're using is to try to look at transcription factor motif discovery and try to identify the transcription factors that might be regulating this transition. Okay, so I'm just going to summarize here. So in the first part of my talk, I told you that we identified this gene kismet in a genetic screen, that it's the flight ortholog of CHD7 in mammals, and that we believe it functions in a coordinated way with trithorax-related complex, or MLL3-4. They co-regulate many genes and co-bind in the genome, and one of the important target genes, at least for stem cell biology in our context, is this gene CBL that's important to limit stem cell self-renewal. I also showed you that we identify 7 major chromatin states within the Drosophila intestinal stem cell lineage, and that transcription factors that control lineage decisions primarily follow this polycomb repressed state to an activated state in the differentiated cells, and that physiology-related genes seem to undergo this transition from a black state to an active state during lineage differentiation. And just one slide, my last slide, just to give you an idea of sort of our broader questions. So we're interested in really understanding the requirements of chromatin changes in both healthy tissues as well as stressed contexts, such as tissue damage or in these cancer-like contexts. So I'm just going to stop here and thank my team. So this work was really spearheaded by Louis Gervais, who's a senior scientist in the lab. Manon did all of the work in the second part of my talk on chromatin state transitions. They had a lot of help, bioinformatic help from Marius van den Beek, as well as Natalia in the lab. We have a close collaboration with our sequencing facility, and also Andrea Brand and Aurora Marshall and other groups. I'll thank my sources of funding, and I'll just thank you for your attention, and I'm happy to take any of your questions.